Bonjour, hello everybody. I'm Hilde, the community manager of the MOOC on viruses and human cancers, and I welcome you all to the third Hangout we have. We have now 3,608 students enrolled in the course from 121 countries. The top 10 countries are France, Morocco, Algeria, UK, Tunisia, Belgium, Mexico, Brazil, USA, Spain, and many, many more. And I want to introduce you to the MOOC director, Jean-Pierre Vartagnan, and one of the lecturers, Pascal Pinault. And we are addressing three questions you have been posting in the discussion platform until today. So the first question refers to telomerase and cancer. In the second question, after the first course of the 7th week, it is said that the telomerase is active only in the tumor cells. But what about its activity in the germinal cells? Thank you for your answer.
So it's the young lad who asked this question is right. In fact, it is true that there is an expression of the telomerase in germline, but in other cells as well. In fact, telomerase is composed of two components: one protein, encoded by the TERT gene, and one RNA encoded by the TERT gene. And in fact, when people are talking about telomerase, they are speaking essentially about the TERT, the protein. And it is true that this gene is active in the germline, in the testis, and in the ovocyte, but in the adult animal, in the adult mammals, it is not active anymore, except in selected small component of our body, like some stem cells in the crypt of the colon, some stem cells in the small intestines, and other location like a stomach or spleen, but at a lower level. In fact, it is reactivated during the tumor process, not with an exceptionally high level of expression, but still it is there, and it contributes to the transformation and the maintenance of the chromosome in the tumor cells. That's what we can say about telomerase.
Thank you very much. The second question we obtained refers to the E6 and E7 genes. The first question is, does the normal human genome contain E6 or E7 genes, or are they only found in HPV-infected cells following viral integration?
E6 and E7 belongs only to viruses, and these two genes are specific to papillomaviruses. E6 and E7 are not belonging to human genes, the first point. E6 and E7s are present in all papillomaviruses, even in mostly present in high-risk papillomaviruses like HPV 16 and 18, but also present in other papillomaviruses like HPV-1. So E6, how you may know and heard during the lecture, E6 binds to the p53, and then the complex is degraded to the proteasome. At the opposite, E7 is binding to retinoblastome, and also it's degraded. So to be quick, to be short, E6, E7 belongs to viruses and not to the human genome.
Thank you very much. The second question was, does the murine leukemia virus affect humans in any way?
So far, murine leukemia viruses, which belong to retroviruses family, do not infect humans. A couple of years ago, there is a controversy on xenotropic murine leukemia virus, saying that probably this virus can be linked to pathology like prostate cancer. So there is, you can have a look in the literature, there is many papers saying that this is now wrong, and probably this suggested link between this murine viruses and prostate cancer is due to contamination. So far, there is no infections by murine leukemia virus in human cells.
Thank you. then we address already the last question we obtained, which refers to clonal expansion and to the phrase, the virus will undergo clonal expansion of the infected cells instead of using reverse transcriptase. According to the slideshow, however, clonal expansion takes place after reverse transcription. So does HTLV1 perform reverse transcription? Please clarify it for me.
This is a very interesting question, and this question is open to the pathology associated with the viruses, with these HTLV1 viruses. If you make the parallel with HIV, HIV is a virus which infects T cells, and it's highly replicative, and the reverse transcriptase of HIV is working a lot, meaning that you have a lot of replication and you have lysis of the cells. So HIV will be never linked with cancer. At the opposite, HTLV1, as you know, could be associated with adult T leukemia, which is a bad cancer. Why? Because these viruses is integrated like HIV, but it's the replication way is mainly done by clonal expansions, meaning that, of course, during the first step, when the virus infects the cells, you need to have a retrotranscription cycle to get the double DNA, the opposite strand, and then integrated. As soon as the virus is integrated, you have less replication because you have less viral particles. So the biology of these two viruses are different, and you explain why you have much more cancer associated with HTLV1 compared to HIV1. Because HTLV1, the mode of replication is mainly clonal expansion. You do not lyse the cell, but you transform the cells. If you compare that to HIV, which can lyse the cells.
Okay, thank you very much. I hope it was clear now. So actually, we are already at the end of our Hangout number 3 for today. The MOOC covered now all 7 weeks by now, but you still have time to attend the weeks, the lectures, do the multiple choice test, and we would be also very happy if you could fill out the evaluation forms we have for each lecture and week. You have the chance to do an authenticated certificate at the end. The time for registration will be announced via email, and the last day of the MOOC will be the 13th of May. If you have more scientific questions, please post them in the discussion platform until then. And if there are enough questions, we will come back to you with a final hangout. So we wish you all a very nice day. Bonne journée et à bientôt. Au revoir.
Bye.
Bye.
Bye.